So I'm sure you have seen the word MERS on the news in the past few years. But do you actually know what MERS stands for? So what is MERS? MERS, or otherwise known as the Middle East Respiratory Syndrome, is an illness caused by a virus, more specifically coronavirus, called Middle East Respiratory Syndrome coronavirus, in short MERS-CoV. This virus was first reported in 2012 in Saudi Arabia. The origin of the virus is currently not understood well, but it's highly likely that it originated from bats and then transmitted to humans via camel as a source of infection. MERS affects the respiratory system such as lungs and breathing tubes. Most MERS patients develop severe acute respiratory illness. About 3 to 4 out of every 10 patients reported with MERS have died. Signs and symptoms of MERS include fever, cough, shortness of breath, and in more severe cases, pneumonia and renal failure. The incubation period for MERS, which is the time between which a person is exposed to MERS-CoV and when they start to have symptoms, is usually around 5 or 6 days, but it can range from 2 to 14 days as well. People with weaker immune system tend to have a higher risk of getting MERS, and the virus appears to cause a more severe disease in older people or those with chronic diseases. MERS is able to spread by close contact between individuals through aerial transmission. Like other coronavirus, it is thought to spread from an infected person's respiratory secretions such as through coughing. However, the precise ways the virus spreads are currently not understood well. People at risk for contracting MERS include those caring for or living with an infected person. Infected people have spread MERS to others in healthcare settings such as hospitals.
To summarize, cells often respond to an invading pathogenic virus by wiring nuclear acid binding to the toll-like receptor that activate the cell nucleus to transiently produce interferon that serve to immunize an affected cell. Through this particular mechanism, the body puts a limit on viral infection, and a more permanent form of defense is achieved by the adaptive immune response. Regarding MERS infection, double-stranded RNA found in MERS coronavirus are recognized by the innate immune system through activation of cytosolic and membranous pattern recognition receptor. Once these receptors recognize the double-stranded RNA, they trigger the activation of interferon regulatory factor, and this leads to the induction of type 1 interferon. Type 1 interferon activate the immune antiviral effectors such as NK cell, T helper cell, and macrophages to pave way for viral clearance. MHC is a cell surface molecule that plays an important role in adaptive immunity. It binds with the peptide fragment derived from the pathogen and displays that on the antigen-presenting cell surface for recognition by appropriate T cells. As for MHC class 1 presentation, antigenic protein are fragmented in the proteasome. The peptide are transported to an ER by 2-type molecule to prevent degradation in cytosol. Stable complex of MHC molecule and peptide is transported to a cell surface for presentation. For MHC class 2 presentation, antigenic protein is degraded into peptide fragments and taken out into intracellular vesicle. It is fused with the exocytic vesicle that contain clip attached class 2 molecule. The aim remove the clip, allowing other peptides to bind and subsequently transported to a cell surface for presentation. At the MERS coronavirus infection site, the antigen presentation of MHC class 1 and class 2 in macrophages and dendritic cell are downregulated, strongly inhibiting the activation of T cell and causes immune dysregulation.
Type 1 interferon expression is a crucial component of this initial immune response, and coronaviruses prevent interferon induction by encoding a suite of genes that antagonize cell-intrinsic innate immune defense programs in the infected host cell. Accessory proteins play an essential role in viral replication and evasion of the host immune response. The MERS coronavirus has 5 accessory proteins, 3, 4A, 4B, 5, and 8B, encoded by various open reading frames. Interferons are secreted by the virus-infected host cells and provide a protective shield to the other exposed cells. Type 1 interferons and inflammatory cytokines are produced because of the recognition of pathogen-associated molecular patterns by RG1-like helicases or TLRs. Proteins M, 4A, 4B, and 5 are involved in the inhibition of interferon production. Protein 4A strongly affects viral pathogenesis. Protein 4A blocks interferon induction and works as a strong inhibitor by inhibiting double-stranded RNA recognition by cellular IgG1 and MDA5. When infected with the MERS coronavirus, infected cells are not able to produce interferon because of the interference present, hindering the binding of double-stranded RNA to RG1-like helicases. The mentioned properties help MERS evade detection by our host immune response. The MERS coronavirus has evolved multiple antagonistic mechanisms to dampen the host defense, which contributes to the high pathogenicity in humans.